let's proceed looking on the properties of the polysaccharide. And another property of a polysaccharide is simply here we can say that the energy storage. So polysaccharides such as the starch in the plant and the glycogen in animals serve as energy storage molecule. So they can be broken down into molecules, or they can be broken down into monosaccharide when energy is released. So most of the polysaccharide, they are used in the storage of food. For instance, kwenye plant they use starch ambayo sasa ina store zile carbohydrate ambazo zinaweza zikause kwenye adverse condition, but also within a plant we have glycogen which are used to store food. And here you can see that most of the polysaccharide, they can be broken down into its monosaccharide when energy it is needed. Kwa pale ambapo panakuwa na starvation within any kind of organism, such as it can be a plant or an animal. So the stored food which are found either within a plant which is in the starch, within animal which is in the glycogen, they are used they are broken down into its monomers or its monosaccharides so that it can release energy in a living organism. But also we have another property, that is the structural role. That is some polysaccharides some polysaccharides like cellulose in a plant and chitin in a plant and chitin in a fungi provide structural support and rigidity. So some of these polysaccharides, kwa mfano ile cellulose ambayo inapatikana kwenye plants ina provide rigidity in the structure, but also the chitin which is found within the fungi pia na yenyewe ina provide the rigidity in the structure of the fungi. So simply we can say that the polysaccharide, mostly cellulose and chitins, they are used to provide the structural support within the those organisms, that is plant and the fungi respectively. And another feature of the polysaccharide here, we can simply say that polysaccharide, they are non-sweet. So unlike polysaccharide unlike monosaccharide and disaccharide, polysaccharide generally do not have sweet taste. So while we are discussing on the disaccharide and the monosaccharide, we have seen that all of them, they are sweet in taste. Lakini sasa tofauti nyingine iliyokuwa kati ya polysaccharide na zile monosaccharide na zile carbohydrates nyingine is the absence of sweetness in their structure. So ukija kuangalia hizi polysaccharide zote tunasema kwamba they are not sweet, lakini zile saccharide nyingine kwa mfano mono pamoja na ile di zote ni nini? Ni sweet in taste. So that is the property of polysaccharide. Another properties is digestibility. Many polysaccharides, they are not digestible by human, such as cellulose, which contribute to dietary fiber. So these properties make polysaccharide essential for various biological functioning and application in the food and the industry. Tumesema kwamba most of these polysaccharides, they are digestible. They are not digestible. You cannot digest the polysaccharide. Kwa mfano kwa mfano cellulose ambayo inapatikana kwenye plants, it's only contributed to dietary fiber, but you cannot digest the fibers which are found within the cellulose. So ile cellulose inasaidia tu kwenye umeng'enyaji wa chakula, lakini they are not digestible, na ndio maana unaweza remove out as a feces product kwa sababu yenyewe haiwezi ikabidi digested completely. Na ndio maana ukila vyakula vingi sana venye kamba unakuta unaenda kutoa as a fecal material with those fibers. Lakini sasa those fibers, they are important because it facilitate or they are contribute in dietary fiber. Inasaidia sana kwenye kumeng'enya chakula. Though haina matumizi mengine apart from that. So tunasema kwamba these polysaccharide, they are undigestible. So all of the polysaccharide, the properties that we have already seen, it make the polysaccharide to be used in various application in the food and the industry due to those features. So now let's see another thing here. Let's start looking on the features of the let's start looking on the examples of the polysaccharide. And now here we have starch. So the starch is the polymer of hexose sugar, that is alpha glucose, that can be extracted as a white powder. It is a major storage carbohydrate of a plant, and it is a product of photosynthesis. What does that mean? Hii starch inatengenezwa na alpha glucose, inatengenezwa na alpha glucose, au inatengenezwa na hexose sugar ambayo inaitwa alpha glucose. Kwa hiyo sasa tunasema kwamba they can be extracted as a white powder. It is a major storage carbohydrate in a plant, and it is a product of photosynthesis. Tuliona kwenye photosynthesis, the final product of photosynthesis is starch, production of starch. So the starch, it tends to be used as a storage organ for a carbohydrate in a plant. And chemically, starch is a polymer of repeated alpha glucose unit that are bonded together by glycosidic bond. So this polysaccharide is a matrix, or is a mixture of two substances, that is amylose and amylopectin. So tunasema kwamba hile formation, tunasema kwamba artificially starch pia inaweza ikatengenezwa. How can it be made? Here simply we can say that it can be made with the repeated polymers of the alpha glucose unit. So zile alpha glucose unit zikiungana zikawa nyingi, ndipo ambapo sasa inatengeneza hii structure ambayo inaitwa nini? Starch. By the production of the glycosidic bond. But also we have seen that the polysaccharide, it is a mixture of two substances, that is the amylose and the amylopectin. So in order for the formation of starch, there must be a production, or there must be the presence of amylose together with the amylopectin. Reminding you, this product, that is amylose and amylopectin, they have been made with a continuous long chain of alpha glucose. So ndipo ambapo sasa zikiungana inaenda kutengeneza sasa hile structure ambayo inaitwa the starch. So amylose unit is a linear helical chain which consists around 50,000 to 20,000 monosaccharide connected by 1,4 glycosidic bond between the glucose unit. Kwa na unaweza ukaona kwa maana kwamba hile amylose group imetengenezwa na about 500 to 20,000 molecule of monosaccharide, that is the alpha glucose. Kwa glucose zaidi ya 20,000 au zaidi ya 500 ndizo ambazo zimetengeneza the first component of the first component of starch ambayo tunaita amylose, ambayo pia inatengenezwa by 1,4 glycosidic bond of a glucose. So tukija pia na hii amylopectin amylopectin differs from amylose, is being highly branched and has short side chain of about 30 glucose units linked by alpha 1,6 glycosidic bond in addition to 1,4 glycosidic bond. We're going to see that in the figure 1.24. So amylopectin molecule may contain up to 2 million of glucose unit. So amylopectin unit may contain several units of glucose. Ndipo ambapo sasa imekuja kutengeneza hii ambayo inaitwa starch. Lakini kuna tofauti mkubwa kati ya amylose na amylopectin. Utofauti wenyewe unakuja kwenye ile bond formation. Amylopectin kuna formation of bond ambayo inaitwa 1,6 glycosidic bond in addition to 1,4 glycosidic bond. So there is two formation of bond. The first one is 1,6 glycosidic bond and 1,4 glycosidic bond. While we are looking on the amylose, we have seen there is only one bond, that is 1,4 glycosidic bond, and thus bring about the difference between the amylose and the amylopectin. So now let's see the figure. You can see this one, the figure which express the structure of amylose and the amylopectin. So unaweza ukaona hii amylose ambayo ni ya kwanza A, this is one amylose. It is a linear structure, ni linear structure, ni kubwa sana. So ukikata hichi kipande kimoja cha amylose, ndipo ambapo unaweza ukaexpress this way. So unaweza ukaona kwenye hii amylose inatengenezwa baada ya kutengenezwa kwa 1,4. This is 1, 2, 3, 4. We have here 4 and here is 1. So now you can see here is just only one. This is only one 1,4 glycosidic bond. Hii ndio only 1,4 glycosidic bond. Now you can see it is quite a difference from this structure, which is the amylopectin. Amylopectin ni tofauti ukikompare na ile ya amylose, ambapo sasa kwenye structure ya amylopectin unaweza ukaona there is a formation of 1,4 glycosidic bond, but also there is formation of alpha 1,6 glycosidic bond, ambayo hii imetokea huku kwenye hii structure. Ukianza kucount vizuri hapa unaweza ukaona this is 1 ambayo imetokea kwenye carbon namba 1. Alafu sasa kuna hii glucose molecule ambapo sasa imecontribute to the formation of 1,6. Started counting from here. This is 1, 2, 3, 4, 5, 6. Ikatengeneza this bond ambayo inaitwa 1,6 glycosidic bond. So now you can see it is very important for you, my student, to know, to notice the difference between a glycosidic bond and between the amylose and the amylopectin in their structure. And by saying so, this marks the end of our today's session. I hope you understand well the difference between amylopectin and the amylose. So thank you for following us at Educare TZ. Keep watching so that you can benefit a lot of series of discussion about cytology and biochemistry. Thank you and bye-bye. Have a nice moment.